I hope you knew what you were doing when you invited me, because I'm no longer a developer. And so this won't be really in the weeds on the technical side. The last time I actually wrote any program of interest was probably 2 years ago when I was trying to cheat at Wordle. Raise your hand if you did the similar thing when Wordle came out. Nobody. My family is right, I am weird. Well, anyway, that was a fun exercise. But I don't do much coding anymore. But I did start my career as a software engineer. And so think of this keynote as a software engineer's view or tour through what's new in the human genome. And hopefully, I kind of think the same way that a lot of you think, and maybe you'll get something useful out of this that you didn't see in kind of the popular press when this was announced in 2022. So the first thing kind of has to do with a comment on this idea of genomic data science, which is kind of what NHGRI branded this new center as. And I kind of laugh at that term because it sounds like the same word. Genomics and data to me are like interchangeable, so it's like saying non-bred or like genomics or something like this. Because genomics is inherently a data science, like DNA is information. Look around you, you're living in the matrix. It's just information everywhere. DNA in all of you, DNA on the carpet, DNA on the walls. It is absolutely everywhere. And just to put it into perspective, each one of you has around 3 trillion nucleated cells. Most of your cells actually don't have a nucleus, your red blood cells, for instance. But 3 trillion left that do, 6 billion bases of DNA in each of them. Each little base pair, if you went in with a ruler and measured it, is 3 angstroms in height. And so you multiply all those numbers together, and it's 4 billion miles of DNA in each one of your bodies, which is enough to stretch from here to Pluto. So have no fear, we will have infinite DNA to compute on with these pipelines forever and ever, because if you then multiply by the 8 billion people on Earth, you get 4.6 million light years of DNA, which goes to the Andromeda galaxy. This is truly astronomical scales of data. So when the Human Genome Project started, they had a much more practical goal of 1 human genome, 1 cell's worth, 6 billion bases. That equates to a meter of DNA. Nonetheless, that 1 meter of DNA took $5 billion to read the first time around. It was launched in 1990, completed in 2003. International effort, ton of money spent, but obviously transformational for this whole field, and we're all reaping the benefits of this early investment in genomics. And it's very apropos that we're at a 20-year anniversary of the conclusion of that project. And here's the final paper out of the Human Genome Project International Consortium, finishing the euchromatic sequence of the human genome. And they were very good about being precise in their language in this abstract. I credit them for that. They said we only finished the euchromatic portion of the genome. And if you read the abstract, they admit there's 300 gaps, and they say that many of these gaps are associated with segmental duplication work with new methods. So that's where we came in 20 years later with this project. But you can understand how the popular press kind of got this wrong. Nobody knows what euchromatin is. I doesn't know what euchromatin is. It's the bit of the genome that has the genes. It's accessible. It's cranking out transcripts. It turns out that's about 90% of the genome. It has had tremendous benefit in genomic medicine over the past 20 years, but there's still that 10% of the so-called heterochromatin and a little fraction of euchromatin that was very repetitive. This is the centromeres and other repetitive bits of the genome that were just excluded. They weren't excluded because they weren't interesting. The centromeres are obviously critical for cell division. The ribosomal rRNA genes that build your ribosomes are in these heterochromatic regions that were left unfinished. So it's like some of the most fundamental parts of the cells were left out in this first reading, and it was a very simple reason. The pieces were too small. So if you're doing a puzzle and it's got a lot of repeats in it, like Waldo here, you're gonna just do the easy, unique parts of the puzzle first, and you're just gonna push the small repetitive bits to the side of the table, deal with it later. That's what happened with the Human Genome Project. The Sanger sequencing at the time was dealing with read lengths of about 500 bases. Then we regressed in the 2000s and went to like 50 base, 100 base pair reads. That was like the dark ages for me as a genome assembly person. But thankfully, we've come back around in the last 10 years to much longer reads. And so the solution to finishing the human genome is a very simple solution. If you're given 3 billion pieces and they're all really tiny, just make the pieces bigger and your job is a lot easier. So we were very happy that around the early 2010s, long-read sequencing or single molecule sequencing came on the market with the initial PacBio instruments, and that got us very excited that we would finally have bigger pieces to deal with. And then in about 2015, Oxford Nanopore came on the market. And this was the one that really blew our minds because Nanopore could do ultra-long read sequencing, in some cases getting a single strand of DNA, 1 million base pairs in one pass, read out with an accuracy now approaching 99%. Just like the astronomical scales of the data in your body, the scales of the nanopore I always like to highlight because it boggles my mind too. This is the R10 nanopore, a little washed out on the screen, but the dimensions are there, about 100 angstroms square. If you scale that nanopore up to the size of my fist and similarly scale the DNA, that's like threading 3 kilometers of rope through your fist and reading off all the bases that they go at 400 bases a second. And that's all done with a tremendous amount of magic on the biochemistry side, the biophysics side, and the machine learning side. All of the base calling now for nanopore sequencing is state-of-the-art neural network approaches to actually get those bases read, and they can do it with, like I said, 99% accuracy with the latest R10 chemistry. So in 2018, with these new weapons in hand, Karen Miga and myself launched this Telomere-to-Telomere consortium with the goal of using this new tech to finish these last remaining gaps. Karen's expertise is in satellite DNAs, and so I was looking at these gaps saying, gee, we should finish these, and Karen was the one that actually knew what was in those gaps. And so our expertise here was really complementary to come together and finish this. Again, we like our simple solutions. We were just going to sequence the heck out of this one genome. And so Shalise Brooks, who's a tech in the sequencing core at NHGRI, spent literally like 6 months because we had this early GridION instrument. It didn't have great throughput. We ran over 100 flow cells of this, just keeping the machine running around the clock to generate, at the time, an astronomical amount of ONT data, maybe 50x coverage in these very long ONT reads. And so this new tech over the last 20 years allowed us to go from the Human Genome Project in the 1990s, where you had rooms full of sequencers. I think this might be the Sanger, I could be wrong, of these old ABI-style sequencers replaced by one tech and one machine generating the data necessary for this project. And then on the back end, I had the very good fortune of recruiting 2 tremendous postdocs to my lab right at the time we had finished generating this data, Mikko Rautiainen on the left and Sergei Nurk on the right. And they came together with a really great approach to combine the PacBio Hi-Fi data and the Oxford Nanopore ultralong data. We started with a PacBio Hi-Fi assembly graph, which is shown here on the left. Each of the little colored noodles is a piece of DNA. If we zoom in on one of the more complex regions, around the centromere of chromosome 2, you can see that it has this tangled structure, as we would call it. Each of the gray rectangles is a sequence of DNA, but there's edges going between them when we're unsure of kind of the order and orientation of those pieces. And so, for instance, this little node here in the middle is a repeat sequence that's adjacent to these 2 unique sequences. We're not sure in which order we should traverse it, so we just keep all of that information encoded as a graph. The Hi-Fi data alone wasn't able to resolve these complex repeat structures. But using Mikko's GraphAligner software, we were able to take those Oxford Nanopore reads, which in some cases were a megabase or more, shown here in red, and align them to these nodes. And this is just basically dynamic programming, Smith-Waterman style alignment, but done in graph space. And you find those nanopore reads mapping to the graph in a very clear path. And so this now gives you an ordering of those nodes. This top nanopore read says, I should start in this node and then go to this node, and now I have a choice, I should go to this node on the right. And that ordering that's inferred by the nanopore reads basically lets you put a number next to each of these nodes, and then you simply follow the numbers 1, 2, 3, 4, et cetera, concatenating those sequences as you go, and you take a very tangly, complex structure and linearize it entirely. Absolutely mind-blowing that this worked. I don't think that any of us really thought that we could finish the genome. It seemed like such an unreachable goal, but 2020, the data was just long enough, just accurate enough that it all came together, and it snapped into one piece in the summer of 2020, and we got all of the chromosomes done, and that was the release of the CHM13 T2T reference genome. And so if we compare the essential stats of the genome released in 2003, which was HG35, with CHM13 in 2023, all of the gaps have been closed. But what I also want to highlight is the reduction in cost is nearly like 1,000,000-fold, which is amazing. And the error rate also got pushed down by 2 orders of magnitude. So from 1 error per 100,000 bases to now 1 error per 10 million bases. And so we've reduced costs by about 1,000,000-fold, increased the accuracy by 2 orders of magnitude, and that's all thanks to the investment in new and improved sequencing technologies and algorithms over the past 20 years. So a testament to a lot can happen in 20 years. This new reference assembly is much more representative of what a human genome actually looks like. So if you pick any sequence out of this new reference and ask, what's the copy number of this sequence compared to any random human on planet Earth, it's 10-fold more likely that CHM13 is going to represent the correct copy number compared to these older references like even HG38, which is underrepresentative of copy number generally due to collapses of very similar repeats, and also inversion status and so forth is much better in these new genomes. So the title of the talk, what's new? We filled in these gaps. What did we actually find? Can be summarized by 2 words, segdupes and satellites. So segdupe stands for segmental duplications. This is a large chunk of sequence, sometimes multiple megabases in size, that's just basically copy-pasted from one part of the genome to the other and highly identical, say over 95% identical. Satellites are also a repeat, but these are the tandem-style repeats where you have maybe 1,000 base pair unit repeated in tandem many, many times, and those tandem arrays make up the centromeres, for instance, and those too can be 5 megabases or more in size. And those segdupes and satellites are highlighted in magenta in this figure on the left, which is showing all of the regions that were filled in with the CHM13 project. And compared to HG38, it is about 8% new sequence, totaling over 200 megabases of sequence. About 3/4 of that sequence are these satellite variety repeats, and about 1/4 of it are the segdupes, which are actually quite gene dense as compared to the satellites, which are free of any gene sequences. But at the bottom, I say an important stat, that compared to HG38, CHM13 has twice the number of bases in segmental duplications that are greater than 95% identity. And so these are very similar repeated sequences through the genome, and as I said, they contain genes and other important sequence. And why this matters is because identity is basically a proxy for evolutionary time. And so because these are highly identical, it means they evolved very recently. They appeared very recently in the genome, and it's precisely these segmental duplications that contain genes that are important for human-specific features, brain development, and so forth, compared to our closest relatives, the great apes. One of the places that is heavily enriched for these regions, as you can see on this plot, are the short arms of the 5 so-called human acrocentric chromosomes. And so this was an area of the genome that we paid particular attention to in the CHM13 paper. And these are known to be functionally relevant because the short arms of these 5 chromosomes are known as the nucleolar organizer regions, and they're responsible for forming the nucleolus, which is a subcomponent of the nucleus. That's basically the factory that cranks out your ribosomes. So here we have this completely essential component of the cell, and the bit that pulls it all together was just completely unknown. The short arms of these chromosomes were just all N in all the prior human references. They're all organized in a very similar way of having a distal satellite region, an rDNA gene array, and these are the RNA genes for the RNA component of the ribosome, and then a proximal satellite region. But in the past, all we could do is look down at the microscope, as we've done here in this fish, and like label the rDNAs, for instance, in pink here. But the exact base for base sequence of all of those short arms was basically unknown. When we finally decoded all of that sequence and looked at what was there, we discovered something that I think is really cool. This is my favorite example from the T2T project and the Pangenome project, kind of discovering new biology. And this was really a discovery that was made in coordination with Eric Garrison, who, if you were in Barcelona, you probably heard about some of this, and Jen Gerten. And what we observed is on these short arms, there are large multiple megabase segmental duplications that are shared between different chromosomes at an identity well above 99%. And so this is sequence on different chromosomes that's as similar as sequence that you would find on 2 haplotypes of a single diploid genome. And that was unusual. We didn't have that in the genome before, and so finding this was a real discovery for us. And the one thing that I want to point out is that, anytime you see sequences that are very similar, I pointed out that that means that they're very related, close in evolutionary time. When you get to like 99.9% and above, we're talking about active recombination in the genome. The reason your haplotypes are so similar is because they're actively recombining via meiosis in the population. And so to find it between 2 different chromosomes was unusual. This led us to the hypothesis that there's actually meiotic recombination happening between different human chromosomes in these spaces, mediated by what we're calling these pseudo-homologous regions. And so these are 2 regions, very identical, between 2 different chromosomes, mispairing during meiosis, getting a crossover event, and in this case of 13 pairing with 21, you're taking the little distal bit of 21, swapping it for the distal bit of 13, and now you have a situation where sequence that was once thought to be maybe unique to chromosome 13 could be found in a different human on a completely different chromosome. And so this kind of breaks a lot of assumptions. A little bit rewriting the textbooks, you learn about homologous recombination. It's always taught the homologs pair, you get the crossover. We know that's not strictly true because X and Y are not homologs. They cross over in the pseudo-autosomal regions. This is kind of analogous to the pseudo-autosomal regions, but on the short arms of the acrocentrics. This led then to a really cool finding that one of those PHRs we found to be inverted on chromosome 14 relative to 13 and 21. And in this case, if you get a mispairing, because that PHR is inverted, you take 2 of the long euchromatic portions of chromosome 13 and 14 and fuse them together head to head, and you get this fusion chromosome that's called a Robertsonian. And this is the most common fusion event in human genomes, probably more frequent than 1 in 1,000 births. And sometimes I'll be giving talks and people will raise their hand in the audience, I have a Robertsonian. It's generally benign, with the exception increases the risk of trisomies or Down syndrome in your offspring. It was always thought that these fusion events happen at the centromeres or the rDNA arrays, but we're finding now it's these PHRs that are mediating, and it's happening during meiosis with kind of standard crossover recombination. So to really prove this, we're in the process of sequencing some Robertsonian carriers to kind of pinpoint exactly the location of those crossovers and look at the haplotypes surrounding them to show kind of the linkage patterns that would indicate a recent recombination event. So that's the biology story and kind of motivating why it matters to have complete genomes. Robertsonians have been known for decades, but we didn't know how they formed. We complete the genome and just instantly, without expecting to find that, we made this discovery by happenstance. What I want to share with you all, though, is kind of all of the other assumptions that break for people that are building bioinformatic pipelines if you want to analyze these very repetitive regions of the genome. And the biggest problem is that our old friend read mapping just fails completely in these repetitive regions of the genome for a number of reasons. And if you want to understand these very recently evolved, highly dynamic regions of the genome, you have to use different approaches. And it really comes down to the idea of reference bias. And so a couple of just very simple examples to show why this happens so we're all on the same page. T to T CHM13 completed gaps. Completing gaps in the reference fixes bias for a very simple reason. If I have a 2-copy gene and one of those genes is absent in my reference and I map a patient to the reference, all of the reads that are originating from that second copy have nowhere to go. They pile up on their homolog, and you get these pile-ups, which are the ugly plateaus in your IGV plots where you get tons of false positive variant calls that basically hides that whole region of the genome from any serious analysis. So obviously, filling in those gaps in CHM13 did a lot to address this type of bias because now the reads can map to the correct location. Here's a real example of that. If you're familiar with IGV plots, this one on the left will give you nightmares. The one on the right looks much better. This is 4 different human genomes aligned to HG38 on the left and aligned to CHM13 on the right over this one pseudogene paralog of the FRG1 gene. There's only 9 of those paralogs in that gene family represented in HG38, when in reality a typical human genome has, say, around 23. All of those extra paralogs just fall on top of the ones that exist in HG38, and you can't call SNPs in this region. But if you align to a complete genome, they go to their correct copies, and you get a much more sane variant pattern. There's a second problem, though, that CHM13 is just a single genome. The copy number between everybody in this room is not equal. These very large gene families tend to vary in copy number between individuals, and gene arrays, where you have the same gene repeated many times, also varies considerably. What I want to point out is that the difference of having gaps in a single reference is really the same as having gaps in the pangenome. If we just have a single reference, we can't address issues where my patient has 2 copies of this gene, the reference only has 1 copy. This manifests kind of exactly like the incomplete reference genome problem. All the reads pile up, you have a problem. You need to map against a reference genome that has the same copy number as the patient that you're analyzing. To kind of highlight a worst case scenario in the human genome that we encountered, we can talk about the TSPY gene array. So this is on the Y chromosome. The Y chromosome is just like a wasteland of genomics if you ever have to analyze it. I'm sorry. There's so many repeats, and there's a lot of reasons why. We can get into it in the coffee break if you're interested. But this is an essential gene for male fertility. It's involved with spermatogenesis. We don't really know what it does. We don't really know any variants in it that have any effect on fertility. And it's because this is what it looks like. Each of these rows is a human haplotype, like an individual assembled Y chromosome. Each of the columns in the little colored boxes is a TSPY gene, a functional gene. And all of the colors are all of the different varieties of the TSPY gene. So think of it as like an allele. And you can see the copy number varies tremendously from about 20 copies to 40 copies. The arrangement of those colors varies considerably. And if you look down at the bottom, HG38 has like, I don't know, 8 copies, and the T to T has 40 copies. So imagine trying to map this individual that has 40 copies onto 8 copies. It's just going to be hopeless. So you simply can't analyze these type of genes with these mapping-based approaches that we've used in the past. There's another even more interesting example on this slide that the far left column and the far right column are TSPY2. And it's a completely separate but related gene, also functional, also protein coding. But in about 80% of the samples, TSPY2 happens downstream of the TSPY array. And in 20% of the samples, it's involved upstream of the TSPY gene array. And there's a big inverted repeat around this array that at some point in the evolutionary history of the Y chromosome, there's been a gene conversion event that moved the functional copy of TSPY2 from upstream to downstream. And so it's not just the copy number that's a problem. It's also that the genes are like moving location, and that if you look at ref-seq annotation or gen code annotation, TSPY2 genes are only named by like the 8 variants that exist in HG38, and so the naming schemes just get completely broken by these gene arrays. But the TSPY2 is an even different problem, that the gene that you care about actually changed location. And so if you think about that happening in my patient sample, the functional copy is the right-hand one, but I map it to a reference where the right-hand one is the annotated pseudogene, all of those variants are going to be misannotated as in a pseudogene and maybe not relevant when they're just showing up there because they were misaligned. And so this is what the pileup looks like if you take nanopore reads. So you think, oh, long reads, this will solve the problem. But I'm mapping nanopore reads against HG38, which has TSPY2 in a different location. The nanopore reads see all of that surrounding genomic context, and they want to map to that. The gene conversion is just like a few thousand base pairs in the middle that's moved it. The nanopore reads kind of outvote that, and they map them all to the pseudogene in this case. And then you get all of this nasty clipping, all of these false positive variant calls, and everything breaks here, and you can't call variants. And this was brought to our attention by Justin Zook of the Genome in a Bottle team because he was trying to make a variant benchmark for chromosomes X and Y based on our T to T assemblies and realized I can't really make a variant benchmark in this particular gene because we can't map reads to it. And so we've learned a lot of interesting lessons in the T to T project about all of the ways read mapping can go wrong. And the first is that structural variation is rampant and probably underappreciated in the human genome because it's specifically enriched in the segdupes and the satellites, which are specifically the regions of the genome that have been dark to us for the last 20 years. The second point is that those satellites in the gene arrays, by nature of them being in these large tandem arrays, evolve differently and by a different set of rules. Rather than just point mutations and small variants, they undergo unequal crossing over events, which expands and contracts the arrays in large chunks. They can recombine with themselves or other parts of the genome. And so rather than just small variant point mutations accumulating, they do accumulate those, but in addition, they have these very large scale evolutionary processes. None of the alignment tools model this. All of the alignment tools, if you think of a scoring matrix for a read map or what have you, they have probabilities for like insertions and substitutions, and that's it. If you try to align 2 centromeres with Minimap2 or whatever your favorite aligner is, it will just completely fail to do it in a sensible way. And it's because the scoring schemes are not set up to model this type of evolution. Segdupes are also super interesting, and the Eichler lab has dug into these and found that they are undergoing a higher mutational rate of small variants, and they're also, as shouldn't be a surprise, more prone to these gene conversion events. By nature of them being so similar, they can recombine with one another in these gene conversions and basically move the functional copy of the gene from one location of the genome to the other. And these segmentally duplicated genes are the ones that are most recently evolved and thought to have an important role in human adaptation. And we know in some cases they're associated with diseases of the type that Evan's lab is interested in, like autism and so forth. And so lastly, because of these weird evolutionary mechanisms, the genes are not always where we think they are. This causes problems when you map long reads. Sometimes you're oblivious to this fact when you map short reads, because they'll happily map to the functional copy and call variants in it without really telling you that the functional copy is now on a different chromosome, for instance. And that location matters, because where the functional copy is determines the regulatory landscape that it's around. And so we want to know where it is in the genome, not just what variants it has. So what's the solution? I don't have a solution for you today. I understand this is a very practically minded conference. And so I can't really say this is how you should all go back and restructure your workflows, but I can kind of give you my vision for where we might be in 10 years. And it's very simply, we all have a unique genome, and we all are our own best reference genome. And when you have a patient come into the clinic, what are you trying to do? You're trying to infer the diploid genome of this individual, and by mapping short reads to a reference, you're just doing a little poor job of assembling that genome. You're getting most of the genes, yes, you're getting 90% of the genome fairly well, but that 10% that Evan and myself and others find most interesting, we just can't do it with read mapping. The flip side of the coin is that, yes, we admit de novo assembly is still costly and requires a lot of expertise. Maybe $10,000 per genome is what we're currently paying for the Human Pangenome Project, with a lot of downstream compute and annotation costs. But I think what we want to do is some in-between of assembly, de novo, and read mapping. Because read mapping is taking advantage of us knowing what a human genome looks like. De novo assembly is basically ignoring all prior information and just saying, just do it from scratch. There must be an in-between. And I credit my former postdoc, Alex Dilthey, I think was the first to coin this term of genome inference in his work with MHC typing. And it's really, I'm looking at these reads, I have a database of other human genomes. Can I infer the complete haplotypes from what I'm looking at, given this prior information about what haplotypes exist in the human population? So think of it as like genome assembly, but with priors. But this presents a chicken and an egg problem, because we need to know what human genomes look like before we can use those priors. And so cue the Pangenome Project, the effort that was launched a few years ago to go out into the population and complete a bunch de novo T-to-T diploid human genomes. And so this is a large consortium, including many centers around the world, primarily organized by University of Santa Cruz, University of Washington, WashU here in the States, but also many others. But what I want to share with you is that it's a very simple idea. If we talk about pangenomes, there's been a tendency in the past 10 years to talk about graph genomes, and that's a very scary concept to clinicians because they don't know what graphs are. They don't know how to analyze graphs. I don't know how to analyze graphs most of the time. But the pangenome doesn't need to be overcomplicated. And I've been winning this mission of getting people to stop talking about graph genomes and just talk about pangenomes as a set of haplotypes. All of the chromosomes in your body are just linear representations of the genome. no different here. We're building a database of linear haplotypes from the human population. And then we want to show that replacing a single reference haplotype with this collection of diverse haplotypes from ancestries all around the world reduces reference bias to a significant degree. And with the right tooling, it yields immediate improvements to the things that we care about in the common workflows of variant calling, accuracy, and comprehensiveness. And a couple of examples that were highlighted in the draft human pangenome reference paper that just came out this summer in Nature, which was a pilot project including about 96 human haplotypes, so a very small representation of diversity, but we still see really nice gains. For instance, a 34% reduction in false positive and false negative small variants when you map with a pangenome aware mapper like giraffe. And giraffe is considering this collection of haplotypes, and it's solving this copy number problem basically and making sure that the reads are mapping to their best haplotype match. They go to the correct gene, and then you project those variants back to the reference genome so you can report it to a clinician in a coordinate system that they're familiar with, like HD38 or even HD19 for that matter. And these precision recall curves are showing this beautiful one over here on the far right is using deep trio as applied to these pangenome-aware giraffe mappings. Not just small variants, but if we talk about structural variants, you can get twice the number of structural variants called, and both of these results are with short reads, by the way. So you can go back to these large short read cohorts and reanalyze them and get some added gain in terms of accuracy and variants called. There's a really cool tool called Pangini from Tobias Marshall's lab from University of Düsseldorf, and twice the number of structural variants called when you use Pangini to infer structural variants. And so kind of like I was just talking about genome inference is actually in the title of the Pangini paper, which I love. They're looking at the pangenome graph, looking at how the short reads map to that graph, and then basically imputing the SVs that must be there based on what they see. And so that gives you a lot of added power. To just kind of give you a flavor of how that works, and it might work in the future long read space. This isn't something that we have implemented now, but I know that Tobias is working on it very closely. I give you this example. So these are 4 haplotypes taken from the pangenome data set of the Rh blood group antigen locus. And so this is what determines whether you have positive or negative blood. And very simply, if you have this one in the top left with this olive color RHD gene, that's a positive. If you are missing that gene, that's a negative. And then down here in the bottom right, you have people that have 2 copies, so positive, if you will, for this RHD gene. And so obviously this is a medically relevant locus, and it's a nice, clean example of the copy number problem that I described to you earlier, that if your reference is the Rh negative haplotype, and then you're mapping any of these Rh positive genomes to it, no variants will be called in those RHD genes. Or if you have this positive, and you map to the reference that has 1 copy, you're going to have the pileup problem, and again, you won't be able to call variants from it. But what if you rethink it into what I was pitching, that we're all our own best reference, and you look at the pangenome and say, for this locus, what's the best haplotype for my sample? You pull out that haplotype from the collection, map to that, call variants against that, thus solving the copy number problem. And I think it will be much easier to infer these haplotypes than you might think. In the middle is the graph structure as represented in the pangenome, and the colored lines are the haplotype path through the graph. And so this top loop here contains the RHD gene. So all of the haplotypes that go through that top loop are telling you this patient has an RHD gene. This one at the bottom that also goes through this little bump on the side, that's the second copy. That patient has 2 copies of the RHD gene. This path that goes through the bottom skips that altogether. This person has no copies of the RHD gene. Those are the haplotype paths. But now think we have 100 kb nanopore reads. You can map those nanopore reads to the graph. They will have a happy way that they go through this haplotype structure. A lot of this variation is common in the population. You can pull out the haplotype that best matches, and then you have a much easier problem. We've got all the common variation recovered in this inferred genome, and now we just have to add the de novo variants from the reads directly. And you can do that with kind of a traditional mapping and variant calling approach. And so I'm high on these methods to, within the next 5 years, really fundamentally change the way that we analyze these genomes in a way that will dramatically reduce bias, which is a good thing because that means all of your genomes will be equally and as accurately analyzed as everyone else's, regardless of how close a match you are to the HG38 reference. So in 2000, when the Human Genome Project was finished, we were talking about draft genomes. In 2010, reference genomes became the buzzword. For better or worse, 2020 made T2T genomes the buzzword. I hope in 2030 we just ditch all the prefixes and we're just talking about genomes, and it's your diploid genome at birth that is basically the basis of all analysis. So the future, I hope, is complete genomes as the basis of analysis. It really is just the most natural way to represent things. Why are we representing everything as diffs against one randomly chosen reference when we can just represent the full context? So I've been working on step one very diligently in my lab to make the routine assembly of these T2T genomes possible so that we can build this pan-genome reference database. I should also credit groups like Hung Lee's that developed the Hi-Fi ASM tool, which has really been the engine of the HPRC over this past year, and that's allowing the construction of these comprehensive pan-genome reference databases to form this prior information of what all of this complex sequence looks like in the population. Given all of that prior knowledge, I think that'll help enable these tools that do cheap and routine inference of T2T genomes. One of the nice side effects of this inference idea is that you could probably do it with much lower coverage. Think of it as small variant imputation, where in the 1,000 Genomes Project, you could get away with doing much lower coverage than you would need for a de novo assembly. Maybe you can do very light coverage of nanopore on a genome very cheaply and get an accurate imputation of the full genome. That then gives each of you your own reference genome. Then the question is what's on there and what matters. I would really love to see ML modeling of the language of the genome, the code of the genome, that switches the model. Instead of taking the genome to the annotation, we're going to bring the annotation to the genome. So imagine a magical ML tool that gets a diploid genome and automatically annotates all the gene structures, the enhancers, the promoters, and at the individual base level, variants of concern. I think this is totally feasible with the right training data, right? So that's what I'm hoping to help nucleate in the coming years with the launch of this center and so forth, to build up that training data so that we can build these type of automated annotation models. And that will then ultimately allow you to serve as your own reference genome. And if you think about any health consequences down the line, if a clinician wants to do a functional study on your genome, or they want to do a somatic variation study, or they want to do a CRISPR design, all of that can be matched to your personal genome, and that will guarantee a reduction in bias and a maximum accuracy. So just to end on one practical note, what can you do today with your pipelines that might take advantage of this? Well, you can, as a first step, try out some of these pan-genome aware tools. These are really being developed to serve as kind of a black box solution so that the user doesn't have to worry about the pan-genome. All of that prior knowledge is baked into the tool. And so Giraffe maps to the pan-genome and reports variants, but reports variants relative to HG38, for instance. But it uses the pan-genome in its data structures to reduce the bias. And so you, for instance, can download the pan-genome 96 haplotypes that we have and run Giraffe on it. It's not that much slower than current single reference mappers. In the SV space, I mentioned Pangini as a really cool tool for doing SV inference. Again, those are both short read tools, but we're working very hard on the equivalent long read tools for similar problems. Consider a personalized reference. If you have a very important functional study, imagine that somebody's working on a cell line, they've got an iPSC line, they're differentiating it, they're getting long-read RNA-seq data for it, they're getting a TAC-seq, what have you. Wouldn't it be great if you could just map all of that functional data to a paired reference that you're dealing with? Or in the case of a personalized genome, you're doing a CRISPR design. If somebody's going to CRISPR any part of my genome, I want to make sure that they're screening those CRISPR probes against the 9% that's missing in the current reference and my own personal genome. And so we've had discussions with the Santa Cruz Genome Browser team. They're going to have kind of a pan-genome aware CRISPR screen tool that when you do CRISPR screens, it's not going to just tell you potential off targets against HG38, it'll tell you potential off targets against all of the haplotypes in this pan-genome collection, which is pretty cool. And it's not that expensive. As I said, maybe $10,000 for a single genome if you want one of these paired references for accurate functional, somatic, what have you. And then lastly, as a room full of engineers, be inspired to go develop this next generation of methods with the goal of reducing bias, because at the end of the day, that's what we're trying to do. Make sure that whoever you are, your genome analysis is tailored to you and is not affected by whatever the choice of the reference genome is. And I would love to see these intelligent ML-enabled tools to bring the annotation directly onto the genome so that we don't have to live in kind of this reference-anchored coordinate system. And finally, it's kind of with Robert's presentation in mind, apologies to all the microbial and plant genomics people that have been doing pangenomics for 20 years. This is my secret sauce to human pangenomics, is that I started as a microbial genomics person, and I used to know a PI named Hervé Tetelin very well, and he was the one who coined pangenomics working on strep genomes in the early 2000s. And so we addressed these problems 20 years ago, and if you want to know how it might shake out in the human world, I would encourage you to go look at how the microbial genomics world has handled pangenomics over the past 20 years, and that has some good lessons to be learned. With that, we practice very open science. All of the resources and tools that I mentioned today are available on our lab's GitHub page. The assembler that Miko and Sergey ultimately designed was called Virco. The initial T2T CHM13 project was largely manual. They took all of those lessons learned and shoved it into an automated process called Virco. If you have hi-fi and ONT data, it can spit out automatically T2T chromosomes for maybe 3/4 of the human diploid chromosome set. T2T CHM13, obviously available with full genome browsers. We recently finished and released a diploid genome benchmark for HG002, which is a diploid T2T genome that is QV75, and we're trying to get that to be the first basically perfect diploid human reference genome. The Pangenome Reference Consortium is aiming to have 700 plus T2T human haplotypes done by the end of the next year. We're currently at about a few hundred. And then lastly, outside of human, we recently finished all of the T2T reference genomes for the great apes plus gibbon, which is going to be a tremendous comparative genomics resource for understanding the complexity and evolution of these very fastly evolving regions of the genome. So I'll end there, and I'm happy to take your questions.
Wow. Thank you for this phenomenal talk. That was very impressive. There's a number of scientific questions that bubble up, a number of technical, practical questions. I have the unenviable job of selecting just the subsets. I am
around for the reception, so
yes.
Feel free to corner me and ask me the hard questions.
I think you might have a lot of customers on that one. I do believe that Venkat is already coding the version of Copilot for ML annotation, so I expect to see a commit very soon. Also, a similar comment/question. Have you started the NF core pipeline for genome inference yet?
We have not. I do not know what Tobias's inference pipelines are coded in. I do have a strong regret that Virco's pipelining system is a bit of a disaster. And so we're going back and re-engineering that in the near future. But unfortunately, no, we don't have a Nextflow pipeline for genome inference yet. That's why it was on my last slide. Please, somebody go do that.
Fair. We might have some volunteers. All right. One question is, so there's definitely short-term, medium, long-term here actions for the community to take. In the very short term, is it worth people switching to the T2T reference, the CH13 reference for their work, or should they wait for like proper pangenome?
So two answers. The first, which I didn't emphasize well enough, is that the pangenome contains CHM13, it contains HG002, it contains HG38. So all of these references, including the T2T references, are in the pangenome. And so if you're using giraffe or pangene against the pangenome, you're already benefiting from the T2T references. And yes, you will get added gain for that. I think that that would be my recommendation over maybe just using a single linear reference, because even though T to T is complete, it's still going to suffer from this representation bias of just being one haplotype. And it's primarily Central European haplotype. The other answer is that if you are interested in understanding the variation within these segdupes, or you're interested in, say, copy number profiling with long reads, then absolutely use the T to T reference. And imagine how much better your copy number profiles are going to be if you're mapping long reads to the T to T reference, and you'll be able to get copy number calling in these segdupes and in these short arms, which are known to kind of have associations and go crazy in cancers and so forth. So copy number calling, analysis of segmental duplications, yeah, the T to T CHM13 reference will give you a much better basis to work in than a GAPI HG reference.
Now, that makes sense. We know that in the community, there's a certain amount of inertia around certain tools. Moving people from 37 to 38 took 10 years, and it's still not finished. There's certain tools like GTK that are used all over the place, do not have support for any of this. How do we move people towards the tooling or get the right tooling in the right places so that a wide number of people can really adopt this?
Yeah, and it's really the source of my pitch to kind of bring the annotation to the genome, because it's really like a liftover problem. We have all of this past information, if you think of ENCODE, NOMAD, ClinVar, there's all of these tremendous data sets that are basically metadata on top of the genome, and they're all anchored on prior genome releases. And so you need to lift that metadata onto the new references if you want to use them, and that's a bit of a hard and tricky thing to do. And so we need those kind of intelligent tools that do that liftover so well that basically then the coordinate system becomes irrelevant, and it's all that knowledge and the metadata getting correctly lifted onto the genomes that's important. And so I don't think that the solution is going to be that we just lift it all over and then we all use CHM13 and we're happy. Like, I'm looking for the next solution beyond that. The reason that I like the giraffe example in the short read space is because that's all kind of doing this and calling variants, and it's able to project them. Because under the hood, the pangenome is a multi-alignment, and we're trying to infer the orthology relationship between all of the bases and all of the haplotypes. And giraffe is aware of that, and it can try in the VG toolkit to map that back to the reference coordinate system where all of your metadata is located on. So we've tried to lift some of these data sets over, but you lift NOMAD over, for instance, and you're only going to lift onto the regions that exist in HG38 because we don't have variants called elsewhere in the genome. So it's a really dicey problem, and it's not going to be solved overnight. And I don't think the solution will be just migrating everybody to CHM13. It's got to be something more complex than that.
Well, thank you very much. I think there's a huge amount of vision there. I'm very excited to see how that plays out. Thank you so much. We are now going to finish. Move on to the reception, which will be in the room just up the stairs on your way out. Reception. One quick housekeeping note before we move there is that for breakfast, it's a very important one. Breakfast will be served tomorrow morning at 8:30 a.m. In a room that's upstairs, but we'll have people kind of guiding you when you come in. Just come into the lobby of the hotel and we'll guide you up to the breakfast room. So please join us there. In the meantime, though, we'll have a fabulous time at the reception. And thank you so much to all of our speakers today, and to Adam. Thank you so much.
Thank you.